Let's first talk about the what's genetic overall. It's mainly a branch of biology, and we know it deals with the heredity and the mechanisms of, let's say, passing the genes through the generations from the parents to the um, offsprings and so on. So it's mainly will describe the mechanism of the hereditary transmission and the variation of inherited characteristics among similar or related organisms. First of all, we have to recall the how the gametes are formed. As we know, it's a production of the meiosis or the sex cell divisions uh, that we will have. It's just that we will have several gametes as a result of this meiosis. Like over here, we will have 4 cells with half number of the chromosomes of the parent or the original cells. So mainly, here's the genetics will play a big role in passing these chromosomes and their genes or the mutations or whatever that are included in these chromosomes into these gametes, and therefore these gametes will make out the new offsprings that will hold these chromosomes. And as we remember that we had what's called crossing over here, that the genetic material will be um, here over here, that the genetic material will be um, recombined between the chromosomes, and it will just pass by the cells until it goes to the gametes. Therefore, there will be some sort of exchange for the genetic material between the chromosomes of the from the original cell. Another thing we have to know is what's an allele. Allele is mainly is one member of the pairs of the series of different forms of the gene. So if we are talking about this whole gene, then we have to know that each gene will have 2 pairs or 2 alleles, one from the mother and one from the father. And if we have like 2 copies or that are identical, so this part, let's say, is identical to this part, then we can say that they are homozygous. While when we will call them heterozygous, then they will have different alleles or different uh, gene uh, that that are inherited from each of the parents. So mainly these are the alleles within the gene of interest. So when we say alleles, you have to always keep in mind that each allele uh, will uh, will be inherited from one of the parents. Inherited patterns are could be monogenic and could be polygenic. Monogenic, mono means one and poly means multiple. So monogenic means that the phenotype is the appearance or the results of the genetic material. Let's say we have what's called a genotype, which is the genetic or the chromosomes or the genes or the alleles that are making the genetic codes, and phenotypes are the appearance or the final production of that gene. So the phenotype over here will be expressed out of a single gene. So let's see here, we have the gene B. If it's capital letter or dominant, let's say, then it will have a purple allele. While if it's recessive or that is abbreviated here in small letter, it will be a white allele. So for the monogenic inheritance, then we are talking about, as we said, one allele, one single gene. So if we have one allele in in big letter and the second allele in big letter, then they will have a homozygous, as we said, but it will be a dominant. So it will have the phenotype or the trait of purple color. And if it's having two of the small alleles, it will have a white color, while in the combination of two, then it will be a heterozygous and it will have a purple color, because as we know that the B will be dominant. We will just discuss it later. And if we are talking about a polygenic inheritance, then we have to know that the phenotype is controlled by multiple genes. Here, as you can see, if it's clear enough, then we are talking about gene 1, gene 2, and gene 3 over here. And each gene has its own combination. So the combination of the three genes will give us a specific phenotype. And over here, another combination of the three alleles will give us another phenotype, and so on. So you can see that here in the skin color, for example, or as you can see the features or the phenotypes of the shape itself um, can recognized by multiple genes. So here it is a result of multiple genes, while here we are only talking about a single. Another thing we have to know that we can consider the inheritance patterns as codominance or incomplete dominance. The codominance is if we have an effect of two or more genes is shown together and on the phenotype. For example, here we are talking about the blood type group. If we have A or B, for example, if it's A plus A, 1A from the mother and 1A from the father, then the blood type will be A. If it's A and O, then the dominant will be A, while over here you can see A and B, then it will be what's called a co-dominance. So no one will be more dominant than the other, while over here the B will be dominant by itself because both of them are from the same, both of the parents have the same allele. So as you can see here, it's called codominance because one or two or more genes will be shown together in the phenotype. Not only one will have the dominance over the other one. The other pattern could be called incomplete dominance because it will be like an intermediate inheritance. Like here, we have a red color that was mated with a white color. Then we have a result of pinkish flower color. So this will be an incomplete dominance where one allele for a specific trait is not completely expressed over its paired allele. So here we said a codominance, so both will be dominant, while here there will be no dominance, but it will be what's called incomplete dominance, where there will be a trait in between the two um, original traits that are mated. You can watch any videos that are here or mentioned so they can clarify the idea from each slide. Again, another thing can be called out of the inheritance patterns also what's called a congenital disease or hereditary or familial disease. The congenital is a disease that present as birth, so the individual will have it once he is born, or a familial disease or hereditary is will be transmitted through the generations, like the Huntington disorders. So this is mainly we have also what's called sex-influenced trait that will be expressed differently between male and female. Like here, the boldness will be expressed more in the males. That you can see that um, a father, if he's a bold, then the probability of having a bold child will be more. So this one is also sex influence. Again, we have what's called a genetic mosaicism, which is a result due the some problem of separation of chromosomes during meiosis. So here it's not mitosis, it's meiosis. We are talking about meiosis. So um, over here you can see that because of the non-disjunction, that here there is like a problem in the separation of the chromosomes that led to a different number of um, gametes as a result upon the meiosis too. So here we have it's like a mosaic. You will have certain cells with different number of um, chromosomes. Like here we have two with one n, here n minus one, here n plus one, there is one extra, and so on. So this is called mosaicism, which you can say that it's like a mosaic or with different combinations. Let's talk about the Mendelian laws. We have this scientist, George Mendel. He conducted some experiments on the peas, and he had nothing to do that time, so he was playing with the peas. So he figured out that it plays a big role in the genetic inheritance, and unfortunately, you have to study this. So, uh, first, first law of the Mendelian laws are the law of segregation. The law of segregation, it talks about the gametes. So, for example, here we have uh, this these production of gametes that each each pair of the alleles of the gene will split, and one gene will pass to the offsprings. So when we talked about the meiosis, we know that the parent cell or the original cell will just pass the alleles to the offsprings to produce the gametes. So this is the first law of Mendelian, which is called law of segregation. We have the second law of the Mendelian, which is called the independent assortment. The independent assortment is mainly states that completely different pairs of allele are passed on to the offspring independently of each other. So, as you can see here, he has this, if you see this dotted shape circle, then it means that it is wrinkled, while if it's like smooth, it is when it's like connected, it is smooth, and each has a different color. So as you can see here, the A is yellow while the B is round. So we have here a yellow round uh, P, while here we have a green. If it's small, it will be the other color, it will be green, and if it's small, then it will be wrinkled. So capital A, capital B is smooth yellow, while small a, small b is wrinkled green. So each of these traits will be independently assorted into the offspring, and you can see that because of this law, you can have multiple combination of the offsprings. Like let's say we will have a smooth green, we will have a wrinkled yellow, and um, if you notice that we don't have a wrinkled yellow as a parent, but within these parent genetic material, you can see that we have the yellow, let's say the yellow color, and uh, the possibility to have this offspring when it's mated. They have the both recessive or the other color or the other trait. So once these are mated, then you can see that these combination can be produced. So this is the second law of the Mendel. Finally, the third one is the law of dominance, that states that the expression of only one of the forms of the parental trace is in the F1 hybrid. Let's see here, this is an example, I've put it for you, that the T is, here the T is talking about the length of the plant itself. So if it's a capital T, a small t, then it will be a big or a tall plant, while here if it's a small t, then it will be a short one. So let's say if we meet these two, then the first generation will be a tall one having the dominant gene as capital T. If we have the second one, you can see that here it will be again tall having these dominant alleles, while it also will have this recessive one with a short phenotype. So you can see that the F1 generation will always display only one of the parent's traits. So as you can see here that uh, here these are the parents, so one of the parents' trait will be expressed mainly in the first generation. Then in the second generation it will be like a combination between the all of the alleles. So to clarify this, you have this video that I have already uploaded it in the Blackboard. You can watch it and you can have a better idea about these principles or law of inheritance. And we have to differentiate between what's called a genetic disease and a molecular genetic disease. Genetic disease mainly means that there is an alteration in the DNA of the genes that will produce a malfunction of the products, the proteins, the messenger RNA. While the molecular genetic disease is like there is a mutation in the DNA molecules that will be inherited between the offsprings. So this is the primary causing event is this mutation. So over here the expression or the product of the alteration will be called a genetic disease, while here it's mainly having the mutation within. You can categorize the genetic diseases into four main categories. First of all, we can call them a single gene, a monogenic disorder, because these are the same meaning as you know, or the Mendelian disorders. Um, Another thing we have discussed is the polygenic inheritance or diseases, which will be a result of multifactorial events that will play a role in expressing these disorders or diseases. We have a cytogenetic disorder. A cytogenetic, mainly we are talking about the chromosome. So they might have some defect in the number of the chromosome or the structure of the chromosome. And finally, we have some disorder that will show atypical pattern of the inheritance. We talked about the monogenic disorders, so mainly it is caused by a single mutation, and it will cause some difference in the structure of the DNA and will lead to some pathogenic consequences. And for example, we have some diseases that will rise from this monogenic disorders, and they can only be understood if we know the biochemical pathways that are affected by these disorders. And mainly, as you can see here, the frequency of these disorders are only 1%. So it is not frequently happening within the populations, and it will follow the typical inheritance patterns as what's mentioned in the first law of inheritance. If you We have these terminologies. We talked about the alleles. The locus is the position. The locus is the position of the chromosomes, of the gene in the chromosomes, and the many locus are called loci. The wild type is the genotype that is happening as a majority in the population, while the haplotype is a set of alleles at a locus or cluster of loci on the chromosomes. A the polymorphism is like if we have from the name poly, it's many. Morphism is from morphology, so it's many shapes of the chromosomes or many, let's say, many alleles or forms of these alleles for the same or the original one. So it is if we have at least two relatively common alleles at the locus in the population, so it is polymorphism. The genotype we talked about is the genetic makeup of the individual, while the phenotype is the expression or the morphology of this genotype. Pleiotropic is a genetic mutation that will cause many effects that will involve different organs. Thing we have to consider when we are talking about the mutation is the pedigree. Pedigree is mainly, it's like a family tree that will show you the history of the family that according to the genetic disorder or the genetic combination within a specific allele. So once there is an individual that is suspected that he have some certain genetic disorder, that he will go for some genetic counseling that they will um, that they will ask for this type of pedigree that will trace, or let's say go back and trace the genetic material of all of the possible parents or sisters or brothers and so on, so they can trace it more clearly. The shape of the pedigree, as you can see here, you have lots of symbols. These 1, 2, 3 is the generation. So this is the third generation. This is the second one. And this is, when you see this arrow, this means that we are talking about individual number 5. He's the one who went and he was suspected to have some genetic disorders that needs to be traced. So they trace the family and they draw this pedigree that says, let's see, each symbol means square over here will mean that this is a male, the circle is a female. Um, if there is a marriage, then you will see this line. If there is a divorce, you will see these dashed lines. For example, if you see some numbers, then the number of the children. If it's colored, then it is affected. Uh, and we said if you see this arrow, then this is the proband or the one who went for the consult. These are the deceased individual or the one who's died, and also some symbols that are important. You might see them in the in the pedigree. Mainly, this is the one you mainly um, find with the arrow. The other ones are like rarely found in the pedigrees. We have what's called the genetic fitness. It's mainly as the quantitative representation of individual reproductive success. So how many successful offspring he can produce. So you can say that he's genetically fit or not. According to the Mendelian law, we can study more the pattern of the single gene disorders or the Mendelian disorders upon two things. Mainly if we have a dominant phenotype or a recessive one, and if it's linked to the um, sex chromosomes, which are the X and Y, or the autosomes, which are the chromosomes from number 1 to 22. So mainly you will have these four categories. If we are talking about dominant, uh, if the disorder is dominant, and if it's related to these chromosomes, the autosomes, so you can call it autosomal dominant. And if it's dominant and it is linked to these six chromosomes, then you can say it is X-linked. On the other hand, we have the recessive. If we are talking about a recessive disorder that is related to the autosomes, then we can call it autosomal recessive, while when it is linked to the sex chromosome, then it is X-linked recessive. So whether it will generate a dominant, recessive disorder, whatever, it will mainly depend or can be determined by two factors: the effect of the mutation that we are talking about, and the tolerance of the biological system to that alteration of that particular gene product. So the effect of the mutation on the function of the gene product will mainly um, result in these four malfunctions that could be summarized into the total loss of function. If we have null allele, then there is no allele, then there is no function. If we have a partial loss of the function in the hypomorphic alleles, hypomorphic means hypo is low, then morphic, we said we have polymorphic, which we have multiple copy of that allele. Hypomorphic is we have less copies of that allele, so we will have less or partial loss of the function, altered function, or novel functions. So let's talk about the how these effects, the what we already mentioned, can be shown within the each of the autosomal recessive and dominant, or the four combination we have already mentioned. If we are talking about the autosomal dominant, so its autosomes, then we are not considering any gender or any XY chromosomes. Then, for example, we have here, if we have the mother is affected, then the affected color will be the blue one. Then if it's dominant, then you will never see a carrier. Keep this in mind. So if it's autosomal dominant, then you will see that uh, the dominant allele will be just transferred to the offsprings, and they might have one, for example, they will have two affected children and two unaffected children over here, regardless the gender or the of that affected uh, offspring. Here we have the autosomal recessive. We are talking about recessive. So for example, if we are meeting two carriers, the carriers will be in sky blue color, then you can see that we have multiple combinations. We will have carrier because we are talking about a recessive trait over here. So you will have carriers regardless the gender again, and you will have one affected and one unaffected offspring. So mainly this is what's related to the autosomal dominant and autosomal recessive. If you see this, let's say this picture, you can see that you can just trace that if there is any carrier or not, and if it's gender linked or not, so you can figure out if it's autosomal recessive or dominant. Let's talk about the X-linked recessive. We said recessive, then you can see that there are carriers. X-linked mainly means that the trait will be held on the X chromosome. So as you know, the offsprings will have the X from either the mother or the father. But if she is a daughter, then she will have a possibility to have one healthy here and one affected. So here she will be unaffected. For example, if she had this X and this X, here she will be a carrier because she had the affected X from her mother, while the healthy one from her father. So she will be a carrier. Here he got, because he is a son, then he will always have a healthy Y, while the X chromosome might be affected from the mother, and in another case, he might have the other X from his mother, so it will be unaffected. Um, so you can see that the X-linked can also have a variety of chances if it's from the mother. Let's see the case if it's X-linked recessive, but the trait is from the it's on the X chromosome of the father. Then as a result, all of the female will have the one copy of the X, but as you can see here, you will not see any affected individual. Why is this? Because they will always have one X from the father, and it will be a mild or a carrier case because the other X will come from the mother, and it will kind of correct the situation that they will never have any affected uh, affected uh, offsprings. On the other hand, they will have always unaffected sons because of the female, because she will give them always a healthy X because she has two healthy copies, while they will always get the Y from the father. So as you can see, the recessive here, they will have a combination. If it's on the mother, then it's more serious than if it's on the X chromosome of the father. Then now the dominant, as you can see, no carriers. Then the dominant, if it's on the X chromosome of the mother, you can see that the same combination of the area will happen, but because if it's dominant, then they will not be carriers, they will be always affected. And if it's from the father, then unfortunately they will always be affected, they will not be carrier even if they have the other X from the mother, because this is dominant, then it will always have the trait, so they will always be having this uh, let's say a trait or disease from the father. So if it's X-linked dominant, then the father will always have a sick daughters, while he will have healthy sons. But if the mother, she's the affected one, then she will be having, she will just pass the affected gene regardless of the gender. Another thing is, for example, we have some cases of, let's say, G6PD, for example. It's a disease that will affect some of the individuals that they will have some sensitivity to the fava bean, that they can't eat it, that it will have uh, some effect on their blood, and so on, because of this deficiency in the G6PD enzyme. So this kind of disease will be if the son is the one who is affected, you have to know that the mother, she's the one who carries this G6PD. It's not the father. So we have these cases mainly happening in our country, that if we have a boy who is affected, then you have directly related to the mother because it's X-linked and it is dominant, it's not recessive. So mainly this is what we have. Combination we have talked about. We said we have the polygenetic inheritance that will be a result from different genes or more than one gene. Plus, we have some environmental and non-genetic influences like the height, weight, mean hair color, for example, that will have uh, an effect of the environment also. And the rate of frequency of this disease is around 2 to 7%.